All right, everybody, we're moving on to chapter 3, cell structure and function. So we're going to be zooming in and focusing on our basic unit of life, the cell. So remember, cells are the smallest units of life. When we did our organization component, we talked about the 6 different levels all the way from the molecular or atoms up to the human organism. So cells were our second tier or second level, but they are our first level that can sustain life. So each individual cell has a metabolism, which is their energy use. They have a synthesis of molecules. They have a means of communication, whether that be via the nervous system, the endocrine system with hormones, um, enzymes. There's some form of communication. And cells are also capable of reproducing. So general cell structure, the outer portion of our cell is going to be known as the cell membrane. We can also call this the plasma membrane. And this structure is what encloses our cytoplasm. And our cytoplasm is a jelly-like substance that kind of holds our organelles in place. Our organelles are specialized structures in cells that have specific functions. This includes our nucleus, mitochondria, ribosomes, endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, cytoskeleton, centrioles, cilia, flagella, and microvilli. So there's a lot of different organelles that make up cells. And we are going to talk about each organelle individually. So this is a look at the big picture of a cell. So you see you have our nucleus here. We have our endoplasmic reticulum. You see a few mitochondria spread around, some lysosomes, our Golgi apparatus, the centrioles, and all of this orange is our cytoplasm with our blue cell membrane. So we talked about the process of differentiation in our last lecture, our chapter 1 lecture. So there's different cell types. So some common types that are shown here, we have bone cell, nerve cell, muscle cells, and red blood cells. And you'll notice that they all look very different from each other, and that's because they have very different functions. So they are structured to perform their specific function. Okay, our first organelle that we'll talk about is the cell nucleus, because this is kind of the most important one as it houses all of our genetic material. So it's usually located close to the center of the cell. It's bound by what's known as the nuclear envelope, and that has both an outer and inner membrane with a little bit of space in between. So that nuclear membrane contains nuclear pores, which are more or less holes where different materials can pass into or out of the nucleus as needed. So when it comes to human cells, our nuclei are going to contain 23 pairs of chromosomes. Our chromosomes are made up of DNA and different proteins. And typically our chromosomes kind of hang out in a type of material known as chromatin, which is just a loosely coiled DNA. But when it's actually time to divide, our chromosomes become tightly coiled, and we can even see them under the microscope when they group together to form a chromosome. So this is the cell nucleus. So this is the nucleus highlighted in our total cell. And then you see we're zoomed in here. We have our inner and outer membrane of the nuclear envelope. You see the nuclear pores throughout the nucleus. You see ribosomes, one of our other organelles, are kind of stuck to the surface of our nucleus as well. And that'll make a little more sense when we talk about those. Okay, so DNA itself is going to contain our genetic information that directs a process known as protein synthesis. And so protein synthesis actually takes place within the ribosome. And this is how we express our genes, or this is how gene expression occurs. So our DNA molecule consists of nucleotides. These join together to form 2 nucleotide strands. They each have a buddy. And these two strands end up connecting and end up resembling rungs on a ladder that's twisted. It's known as a double helix structure. If you remember back when we were talking about nucleic acids, when we talked about our organic material, each nucleotide is a 5-carbon sugar. It has a phosphate group, and it has a different nitrogenous base. So we have 4 different nitrogenous bases. Each nucleotide on one DNA strand is going to have a specific bonding pattern. So it's going to have a specific partner that always matches up. A gene is a sequence of nucleotides that provides a set of instructions for making a specific protein. So our genes are the recipe for specific proteins. So this shows our tightly wound chromatin into a chromosome. Chromatin itself is much loosely wrapped, and then we have our DNA, or that ladder-like structure that's twisted. Okay, ribosome components are produced within the nucleolus, and so our ribosomes are where proteins are actually produced. Our ribosomes are what can read the instructions on our genes and actually make the protein that it's told to make. So sometimes they're attached to other organelles. So one example of this is our rough endoplasmic reticulum. Ribosomes not attached to the organelle. Some of them are just floating within the cytoplasm. Those are known as free ribosomes. So this diagram just shows how a ribosome itself is made. So there's nothing too much to look into on this one. It's just showing that ribosome components go into the nucleus and nucleolus, and it creates our ribosomal units, which become a ribosome to translate the mRNA. The endoplasmic reticulum is really just a series of membranes that form kind of sacs and tubes. This extends from the cytoplasm or the interior all the way to the outer nuclear membrane. There's 2 different types of endoplasmic reticulums, or ERs. The rough ER is called the rough ER because there are ribosomes all over its surface, giving it a rougher appearance. Therefore, the smooth ER has no attached ribosomes. The rough ER is involved in protein synthesis, which makes sense because it has ribosomes all over its surface, which are involved in protein synthesis. The smooth ER is for lipid synthesis, cell detoxification, which means it's a way to package and get rid of wastes. And it also stores calcium ions in skeletal muscle cells, which is quite important in the process to allow skeletal muscle contraction. So you see our rough ER, all these folds of the membrane that create these kind of tubes, the ribosomes on the surface. And then the smooth ER, same kind of membranes, but no ribosomes on the surface anywhere. The Golgi apparatus, also known as the Golgi complex, also is a bunch of closely packed stacks of curved membrane. And it's really membrane-bound sacs, also known as vesicles. So the Golgi apparatus's job is to collect, modify, package, and distribute different proteins and lipids manufactured by the ER. So in a way, you can think of the Golgi apparatus as the post office. They're kind of getting the packages from the ER and then shipping them out to where they need to go. So sometimes they form, like I said, sacs known as vesicles, which some can actually be secretory vesicles or will secrete material into the external environment. You can have lysosomes, which we'll talk about in a little bit, and there's a few other kinds of vesicles, but we're not going to get into a lot of specifics on those. So this is our Golgi apparatus here, and you can see it can kind of pinch off these vesicles or bubbles of material. So lysosomes are membrane-bound vesicles, just like these 2 green circles or vesicles here. They can contain a variety of different enzymes that will function typically for intracellular digestion, or to digest certain materials or dissolve certain wastes. So vesicles are formed by this process known as endocytosis, and they can fuse with lysosomes in order to break down material. There's a little diagram of that next. So one example of this would be when a white blood cell phagocytizes bacteria or eats a bacteria. So imagine this is the cell membrane of a white blood cell, and these little purple dots are some kind of bacteria, some kind of invader that's not supposed to be here. Our white blood cell forms a vesicle, it brings it into the cell. Our Golgi apparatus produces a lysosome that has some kind of enzyme that's going to digest this material that our white blood cell just brought in, and it's going to form a waste. And so that's either going to go and be released to the external environment again, or it'll go somewhere else in the cell to be processed. Okay, peroxisomes are another special type of vesicle. And so peroxisomes are specific to breaking down fatty acids, amino acids, and hydrogen peroxide. And hydrogen peroxide is really a byproduct of our fatty acid and amino acid breakdown, but it can be toxic to the cell. All right, mitochondria. So some of you may have heard the corny saying, mitochondria is the powerhouse of the cell. And so these are our organelles that are responsible for producing a lot of our ATP. And this is done with aerobic metabolism, meaning that we have enough supply of oxygen to make this happen. So they have both an inner and an outer membrane with space in between. The outer membrane has a nice smooth contour, but that inner membrane has a bunch of folds, and these folds are known as cristae, and they kind of project from the outside in on the mitochondria. So it makes sense. Mitochondria produce ATP or energy, and so cells that are going to need more energy or be working all the time or working more often are going to need more mitochondria to be able to supply that energy. So mitochondria is a plural word. It's a single mitochondrion. So you see this outer membrane is kind of nice and smooth all the way around, forms a little oval there. And then this inner membrane has these folds known as cristae. And in between, this middle portion in between the two kind of rows of cristae is known as the matrix. All right, cytoskeleton is more of a category than it is a specific organelle. So there's a few different types of cytoskeleton, which give internal framework to the cell, and also can be structures on the outer portion of cells. So it typically consists of protein structures that will support the cell. They help hold organelles in place and sometimes enable the cell to change shape. So think skeleton, how our skeleton gives us shape and form. Cytoskeleton is the same thing on the cellular level. So a couple examples of this are microtubules, microfilaments, and intermediate filaments. Okay, first we'll talk about microtubules. So these are hollow structures formed from protein subunits. The microtubules perform a variety of roles. This includes helping to support cytoplasm or give the cytoplasm structure. They can assist in cell division. They help form essential components of certain organelles such as cilia and flagella, which are two different types of external structures. Microfilaments also give structure and support to the cytoplasm, and they help determine cell shape. Some microfilaments can be involved with cell movement, kind of projections to help the cell move. And microfilaments in muscle cells enable our cells to shorten or contract. So actin and myosin, the microfilaments are the ones that are in muscle. So intermediate filaments, these are just structural filaments. They're really proteins that just give the cytoplasm support. You see the microtubules here, microfilaments. There's not too much else that helps you out. Just remember that our cytoskeleton components give structure and support. They help determine cell shape. And we can have some external projections that have different roles. Okay, centrioles. So the centrosome is a specialized area of cytoplasm. This is found very close to the nucleus where our microtubules actually form. So it contains two centrioles, which are typically perpendicular to each other. And then each centriole is a cylinder, and it's composed of microtubules. And so centrioles are important in the process of mitosis or cell division. So you see all the microtubules formed around, and this forms our centriole. So cilia can project from the surface of certain cells, and so they're responsible for movement of materials over top of cells, typically. An example of this is mucus. Cilia are cylindrical, and so they extend from the cell and are also composed of microtubules, but they're almost like little, almost like brushes, little brushes. Flagella are somewhat similar to cilia, but are typically longer and usually only one per cell. So the most common example of this is a sperm cell has the one flagellum, or more or less the tail, and they're typically used for propulsion of the cell itself. Our next example here is microvilli, and these are specialized extensions of a cell membrane that are also supported by microfilaments. They're not actively moving like cilia and flagella, but the main job is to increase the surface area. So think of them, I think we have a picture here. So think of microvilli as little hills all over the top of a cell. That really increases the total surface area of their cell membranes. So in an area like the kidney or our small intestine, where there's a lot of absorption happening, having these microvilli structures is really important for them to be able to absorb as many nutrients as possible. Okay, I think we are going to go ahead and take a pause here, and we will continue on with the cell membrane and things related to the cell membrane in the second half of our lecture.